Hi. So in previous session we have discussed a lot about the antigen, properties of antigen, and what kind of this antigen is can induce the immune responses in terms of B cell and T cells, and how these B cells basically recognize these antigen, and T cells also how they recognize, what kind of interactions are there. So now let's move on to the antibody. In previous session, I have discussed about how to generate the antibody, that is two different kinds of antibody, the polyclonal antibody, which is extremely simple, it's very cheap, but generation of monoclonal antibody is manifold expensive, very difficult, and it's technically very challenging in order to make the monoclonal antibody, right. Now let's move to the antibody. Here I would like to discuss about after the conceptualization of that there is some humoral component. Okay, you remember the history of immunology. There are two line of or two school of thoughts. One is that one school of thought is that some cell is involved in defense, okay, and another school of thought is some soluble molecules are involved, or soluble substance is involved in defense, okay. And if you remember, you probably remember that Metchnikoff is the key person who is basically initiated this concept. There is some phagocytic cells which can phagocytose the antigen or microbial pathogen, and there is a school of thought where they have explained, you probably remember that name, Paul Ehrlich. He is a key person in developing that concept, okay. And these school of thoughts are known as a cellularist and humoral-arist, okay. So let's begin with that thing. So he is a Paul Ehrlich, and one bearing, these are the key person who are postulating this or showed by experiment that there is some soluble factor, and these soluble factors are involved in defense, okay. You may remember that his work, Paul Ehrlich, his side chain theory, okay, cell makes some side chain, and then this basically bind with the antigen, and this is present in soluble form in blood, okay. So this side chain theory and this the serum toxin work by Emil von Behring, both of these workers received the Nobel Prize for their contribution, okay. Now there was a after this this work, now people wanted to know what kind of molecule it is, okay, how this molecule work, okay. So in order to understand how this molecule work and how this function, you need to understand first you need to find out whether this molecule is present or not. And this work was very elegantly presented by one or two people. These people basically very nicely showed that, okay, there is a some molecule or some protein is there which is giving this kind of protection, okay. And this was these works was initiated after 1901, okay, and people were giving several theories, but no one are able to convincingly show this thing, okay. So the serum, you know that since late 19th century, the antibodies are in serum. This was kind of established because of the work of a previous worker, okay, like Paul Ehrlich and amyl 1 bearing. Okay. So they understand that this is present in the serum. Okay, but how to find out and what to do it. So that work was further done by another people, which I am going to show you in a short while. So serum is a fluid phase that remain after the blood clotting. So here I just want to tell what is serum. Okay, serum is just simply you take out the blood, allow it to clot at room temperature, and the fluid which is remain in the in clotted blood, okay, over the clotted blood, we call it as a serum. There is a one more contrasting term, just for your information I am telling what is plasma. So plasma is nothing. So you take out the blood and put the anticoagulant. So you collect the blood in with anticoagulant, then blood will not clot. Okay. So if you spin that blood, you will have a straw color fluid, which we call it as a plasma. Okay. So the difference is very simple between serum and plasma. So in case of plasma, all blood clotting factors will be remain there. In case of serum, there will be no blood clotting factors, okay, like fibrinogen and all those things. Okay, you probably studied in earlier of your education. Okay. So antibodies are also found in other secretion. Okay, this was also kind of established by these workers, and this work was basically a very simple work, and this was basically given by these two people, that is Arne Tiselius and Ivan Kabat, or Tiselius and Kabat experiment. This is quite famous, and they performed and they reported in 1939. This is a very simple experiment which clearly demonstrate there is some protein molecule which will be involved in defense. Okay. So now I will explain this experiment. This is very simple, you should understand. Okay. So what they have done, they have taken the defined antigen, or say for example BSA, I don't know precisely what protein they have taken, but I am giving the concept. So they have taken the protein antigen and challenged the animal, most likely they used the rabbit. Okay. And after challenge, they collected the blood after 14 days or in at particular duration, and then they have isolated the serum. Okay. So after challenge, they collected after some time they collected the blood and they prepared the serum. Okay. And when they have prepared the serum, after that they divided this serum into two portions, two equal portions. Okay. It's simple, right? And one portion, what they have done, they have put the antigen in insoluble form. Okay. And they allowed it to react. Okay. So what will happen? You can understand very simply. When you prepare the antigen in insoluble form, it's very simple. You just tag this antigen with some insoluble matrix, like beads, some beads like agarose bead or something like that. Okay. And when you will put this antigen and bead in the serum, in one portion of serum, so what will happen? There will be antibody against this antigen, right? So this antigen will react with antibody which is present in the serum, and once you will do this process, after that if you spin this serum, okay, spin the serum which contain these beads, antigen beads. Okay. So what will happen? All antibody will be removed. Okay. So then after that they have performed a simple electrophoresis experiment. Here you can see that there is a gamma portion. Okay. So we also call it as a immunoglobulin, antibody as a immunoglobulin. We also call it as a gamma globulin. Okay. So this gamma globulin is basically here you can see that. So then they have performed the electrophoresis of both. One is treated, the serum which is treated with antigen and bead, and another is just whole serum. Okay, and then they can find out that, okay, this is a reacting portion, the gamma portion is basically reacting. Here you can see the gamma portion is reacted and removed from the serum. Okay, so in that way this antibody was discovered. Okay, it means the molecule, people can see that, okay, this molecular weight molecule is responsible for the defense. Okay, after that there was several challenges, and the one of the most important challenges, what is the molecular composition, how big, what is all those things. Okay, so this molecular composition of antibody was again deciphered by a lot of people by very simple experiment. At least by this way they can tell that the antibody molecule is consist of that many polypeptide chain. Okay, so what is the molecular composition? So antibody is basically now we know that antibody is basically it's a heterodimers. There are two heterodimers which combines, and that makes an antibody molecule. And this antibody molecule is basically consists of two light chain. I will show you in a short while. And each light chain is basically consist of 22 kilodalton polypeptide chain, and it has a two heavy chain which is consist of 55 kilodalton each. Okay, so how this, so this is basically giving an idea that, okay, the antibody molecule is having that molecular weight, but this is not giving any idea about how they are arranged. Okay, so in subsequent experiment people have deciphered how this molecule looks like. But before that I just want to show you the structure, so that you will be easily understand how this work result to the determination of structure of antibody, and then the functional aspect of antibody. Okay, so the first 110 amino acids in both light chain and heavy chain towards N terminal, they are basically variable, and this depends on antibody to antibody, and the antibody specificity depends on this 110 N terminal 110 amino acids. Okay, so here I am just giving you the structure. I have discussed this structure earlier, but I quickly showed to you when I was discussing the classical complement. Okay, here you can see there is a here you can see there are two light chains. Okay, and here you can see that there is an N terminal and there is a C terminal, and in light chain you can see one towards N terminal there is a darker region, and towards C terminal there is a lighter region, if you see in color code. So this lighter region is the constant portion of it will remain same. Okay, it depends on the kind of light chain. Okay, and here you can see that there are black strips over this towards N terminal in darker region. These regions are basically hypervariable region. Okay, and this light chain is consist of about 214 amino acid. There is a heavy chain. Here you can see there is a heavy chain, and this has also the darker region which is variable region, and there are CH1, CH2, CH3 region. Okay, so these regions are basically constant region. Okay, and now here you can see in the structure in both light chain and heavy chain there are lot of disulfide linkages. Okay, and this disulfide linkages basically makes a very unique structure which we call it as a immunoglobulin superfamily structure. Okay, so immunoglobulin, this makes this disulfide linkages makes the immunoglobulin superfamily structure. And this heavy chain is basically consists of approximately 446 amino acid, and this light chain and heavy chain, this is a heterodimer, okay, and this heterodimer again make a bind with another heterodimer, and then that makes a functional antibody. And these two heterodimers are basically joined by the several disulfide linkages, okay, here you can see. And the black strip which I told you, that this we call it as a hypervariable region, or CDR, we also call it as a CDR, complementarity determining region. This is closely associated with the antigen, okay. The portion which is binding with the antigen, so this hypervariable region or the darker color region in both light and heavy chain, basically they interact with antigen, and we call it as a antigen binding region, okay. In short form we call it as a FAB, the fragment antigen binding, okay. There is a the constant region, we call it as a FC region, and this FC region is playing very important role in various biological activity. I will tell you what is these biological activity, or more precisely the function of antibody, okay. This constant region is also heavily glycosylated, okay. So gamma globulin or immunoglobulin is highly glycosylated molecule, okay. Here you can see that the CHO is basically representing the glycosylation throughout the region, okay. What are the effector functions of this antibody? You know some. One is opsonization. So once the antibody is generated against any microbe, then this antibody will coat these microbe, and then it will be readily phagocytosed. It is not like a complement thing. In case of complement, the microbe is basically coated with C3b or C4b, okay. So the term is same, opsonization. So opsonization means the coating of this microbial pathogen with particular molecule. Here it is antibody. In case of complement, it is C3b or C4b, okay. So antibody has one important function, that is opsonization. Another is complement activation. I have discussed in a great detail, so I am not going to talk any about complement activation. Basically it activates classical pathway, okay. Antibodies are also playing very important role in antibody dependent cell mediated cytotoxicity for killing of the target cells, okay, virally infected target cell. And ADCCs are basically triggered by NK cells, okay, if you remember. The soluble antigen bind with antibody, and this basically makes a immune complex, okay, and this immune complex is basically insoluble, and it can be phagocytosed, it can be removed in the kidneys and all those places, okay, and then this will keep the system clean, okay. So these are the some key function. Besides this, the antibody has a different constant region, and each constant region has some specific function. If you remember, I have discussed during this course, there is some FC gamma receptor, FC epsilon receptor. So FC gamma is basically there is one class of antibody IgG. So IgG bind with FC gamma receptor, okay. FC epsilon receptor, if you remember, I discussed a little bit about the hypersensitivity. Over there is a FC epsilon receptor, okay. There are high affinity, low affinity, okay. So Fc epsilon is basically IgE is interacting with Fc epsilon receptor. So these are some simple, and they have some particular function. Okay. So this is the structure of antibody. This is not precisely true for all antibodies. This is one standard antibody molecule which is close to the IgG, okay, immunoglobulin G. Now I will talk how the structure was kind of determined by simple biochemical method. So this structure was determined by very simple experiments using electrophoresis and by generating antibody molecule against particular fragment. Okay. So this structure was determined by simple method. Here you can see that if you digest this antibody purified gamma globulin, okay, with pepsin, so then it will generate. So here you can see that the intact antibody molecule has a molecular weight of 150 kilodalton. If you digest with pepsin, then it will generate the FAB2 fragment. Here you can see that the disulfide bonds are there, and both light chain and heavy chain heterodimer are still in joint form. That will generate the 100 kilodalton fragment. And if you perform this papain digestion, then you will see there will be FAB fragment, and there will be a FC portion. Okay. So these fragments can be determined by using a specific antibody. I will tell you in subsequent slide. And if you treat this antibody molecule with some reducing agent like beta mercaptoethanol, okay, then you will get heavy chain and light chain. Okay. So this is a, if you see this simple experiment, and this is a kind of puzzle. Okay. If you do this thing, then you will get this kind of thing. If you do this thing, then you will get this kind of result. And when you put all these results together and think, then you can find out the structure of the tentative structure of antibody. Okay. At least how these polypeptides are organized and how they are present in the molecule. Okay. And after that this can be easily detected by simple electrophoresis. Here you can see that there is electrophoresis electrogram, and these in these electrogram they have detected these particular fragments like 100 kilodalton or this 45 kilodalton and 50 kilodalton by simple by making the antibody against these molecule. For example, if you have a rabbit antibody, if you take this antibody from rabbit and inject it into the goat, then you can generate the antibody against that antibody. Means you can generate anti-rabbit immunoglobulin in goat. Okay. And you can use that antibody in order to detect. Okay. So this is the method by which this complicated puzzle was solved, how this polypeptide chain is basically arranged. Here I am giving you the key points how this experiment was performed. They basically immunize immunization of goat with antibody from rabbit. As I have told you earlier. Okay. The immunization with Fab fragment. So you need to isolate these fragments. So at that time electrophoresis and chromatography was quite well established. Okay. They don't have had a very sophisticated techniques and methods. Okay. Interaction with both heavy and light chain. So if you make an antibody against say for example Fab, as you can see the 45 kilodalton, the antibody which is generated against that Fab, this can also react with FAB square. Okay. As well as this can react with heavy chain and light chain, okay, after treatment with mercaptoethanol. So this is a kind of puzzle you need to think, then you can understand very well, okay. Immunization with Fc fragment that also generate some antibody, and that you can see this interaction that is interacting with only heavy chain. Sequence of antibody required a purified source of antibody. Okay, here there is a one challenge. Generally, antibody, when you challenge the animal with antigen, you will get the mixture of antibody, and if you use the starting material as a mixture of antibody, that is very difficult to proceed. So from where they get the purified antibody? The simple answer is they got it from cancerous cell. They induce some cancer in the animal, and that animal will produce only one kind of light chain, heavy chain, or one kind of antibody, okay. So they use that antibody, okay. So B cell from multiple melanoma patient, in case of human, they also use this B cells from multiple melanoma patient, okay. And by doing this different digestion and by chemical treatment, they could determine the structure, okay, the structure as it is shown over there, okay. This FAB2 is still show the antigen binding capacity, okay. After all this digestion, this has a capacity to bind with antigen. So in that way, the structural information was or a structural insight was gained, and of course later on the crystal structure and all those things were there, okay. So with this I will stop here, and in next session I will discuss about various kind of antibody and how this antibody diversity was generated. It's a very complicated, molecularly it's a very complicated, but I will try to give you at least a snapshot about how this antibody diversity is generated. Thank you.